the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Hello, and welcome to the Dementia Researcher podcast. Today, we have a slightly different episode because it is our Christmas edition. And today, we are doing something we do not often do in research. We are admitting that things sometimes go wrong in a show that we've titled The 12 Research Fails of Christmas. I am Dr. Fiona McLean, and I am a Principal Investigator and Alzheimer's Research UK Fellow at the University of Dundee. And in this show, we are going to look back on the year and share small research blunders, lab mishaps, near misses, awkward moments, and quiet failures that taught us something, even if at the time they just made us sigh. Nothing catastrophic and nothing career-ending, just the sort of things that happen when you're doing real research with real humans and real data with real deadlines. I'm joined by three brilliant researchers today who were brave enough to agree to this. We're going to start with Soraya Meftah from the University of Edinburgh. We also have Ian Harrison from UCL, as well as Kate Harris from Newcastle University. Hello, everyone.
Hello.
Hello.
Yay. So before we get festive, could I just ask each of you to do a brief introduction in your own words? And we can start with Ian.
Hello. So my name's Ian Harrison. I'm a Principal Research Fellow at University College London, and I'm an Alzheimer's Society Dementia Research Leader Fellow here at UCL.
Fabulous. And let's go to Soraya next.
Hello, I'm Dr. Soraya Meftah, based at the University of Edinburgh, and I am an incoming Race Against Dementia ARUK funded fellow.
Yay!
That's
Congratulations.
amazing.
Thank you.
And last but not least, Kate.
Hi everyone. No fancy fellowships here, but I am a senior lecturer in interdisciplinary drug discovery at Newcastle.
Amazing, Kate. So before we start counting down our festive fails, I just want to say this clearly for anyone listening, that research does not look like the methods section of a paper as much as we want it to. It often looks like things breaking, plans changing, wrong assumptions, and moments where you realise you should have checked something much earlier. So this episode is really about permission to laugh, permission to learn, and permission not to be perfect. Let us begin. So first of all, let's talk about things that seem obvious, but were absolutely not. So Soraya, I'm going to start with you. Can you tell us something about in your lab that has happened this year where you thought, oh, well, this is so obvious, and when you were planning it, it seemed so straightforward. But then once you started, you were like, oh no, I've maybe not thought this through enough.
I mean, I think when we were prepping for this podcast and when I was reading through the kind of bits and pieces, there's so many, I never really see them as mistakes. I just see them as like lessons to learn from. And I've had a lot of lessons to learn from. So my one for this was actually, I was co-supervising a student this year, my lovely master's student, Millie. And we designed this project for her in mind, and we were like, we're going to look at tau and we're going to look at this. Very straightforward. It's been done in the literature, which is always the first warning sign. We're like, oh yeah, we'll just replicate what's been done in the literature and then we'll extend out. And
how often it's been replicated in literature is you find this new paper and you're like, oh wow, this really new shiny technique, let's do it.
This isn't that shine. This is me being like, this is a very common thing. Like, I'm sure we'll be able to do it in this. It's a different system that we're using. So I was like, yeah, this will be fine. Pilot set of experiments, everything went the opposite direction to what we expected. And then like the next set of experiments, everything went completely the other direction as well. So we had to do a very big pivot. I think it's going to be really interesting. We're still going to get some really cool stuff from it. But the original like hole that we'd kind of settled down and that she'd done all her literature preparation on was not the hole that we ended up going down. It was a very good learning lesson for science.
Amazing. So, Soraya, you obviously learned a lot when that was happening. I always say to my students that you'll learn more from your failures than you will from your successes in the lab. If
Yeah.
everything goes really smoothly, then it's like you never really learn stuff. You always learn when you need to troubleshoot a protocol and things like that. Did it work out, the experiment in the end? Sorry. Did you get data from it?
I think the main thing for me that I was telling her, so this was obviously a learning lesson for both of us, but the main thing I was saying to her was that as long as the experiment has worked, the result is obviously not like we can't control the results. So I watched her do some stuff, and like there was also some other bits that I showed her completely wrong. So again, big mistake on my part. But we troubleshooted all that. The result was still the same. So then we're just following the interesting story that the data is telling us.
Amazing. So when did you realize something was off? Was it end of experiment, midway through?
It was more like I'd planned this thing, so it was kind of like a culture experiment. So then like after 2 weeks or 3 weeks or something, we were then looking to like do some molecular characterization, and we looked at the molecular stuff, and I was like, oh, this is completely opposite what we were expecting and completely not where I thought this was going. But yeah, it's a learning story. It's a learning fun.
So there's not really much to fix. It's more just that's what the data is showing.
Yeah, exactly.
Yeah, but sometimes I think those are the most exciting experiments. Like I did a big single cell experiment, and it's not that it didn't show me what I expected, but there was like just this whole other avenue of research that opened up, and that's what I'm pursuing now. It's what I plan on writing my next fellowship on. So I think sometimes the most unexpected results can actually be the most interesting. And like, did you, when you were kind of just having a laugh with the student about it? Because I do feel sorry for students because they are they have a certain amount of time to collect data for their thesis. So I feel like sometimes when things don't go to plan in the way that they thought they would, they can panic. Yeah, definitely.
Yeah, I think there was a bit of stress, but like I said, we sat down, we made sure she was doing everything correctly, and she was. So, it's just, I was like, it's more for your discussion. It's more to talk about.
Amazing.
Those sentences are brilliant.
Classic.
You start whipping them out. It's like a toolkit of things you've learned, tried and tested that don't make the situation worse.
So, has anyone else had any students, like have they had an impact, students just having difficulties with pivoting experiments?
Is that not every day?
Yeah, I think so.
And I don't mean that as difficulty with students. My students are delightful and I'm grateful for them every day, but I can't remember the last time an experiment went to plan. There's always something.
I feel for students. Like I said, when they've got these time constraints and they're having to sort of, deal with that pressure as well as the pressure of just science research in general.
That's kind of in science though, right? Because I guess people come into as like an MSc, for example, thinking, I guess they've only read papers that appear as if everything works first time. Do you know what I mean? When you come into it and you're like, actually, that didn't work. So, but this is the data. This is the data.
Yeah,
it's not...
exactly.
I'd say, 90% of the time, I'd say experiments don't work. And then it's 10% of the time they do. But as long as the 90% is not, anything too disastrous, as long as it's just, tweaking and taking steps forward. I had a master's student who we were doing a technique called seahorse, and we had a protein inducible cell line. And the thing that you use to induce the protein to be made actually affects respiration. For those of you who don't know the seahorse technique, it
sounds
looking
great.
at cellular respiration. So his master's thesis ended up being on how, if anyone in the literature, if they were using anything that used it was a TET inducible system, actually the seahorse is probably showing that rather than anything else.
The panic though. The panic.
He did so well though, because he was able to talk about it.
Amazing.
And that is what research is. But it did make me a bit concerned about half the literature using these systems. I was like, what are you really looking at?
We had a similar thing. I wasn't involved in the study, but there was a study in my department where they were doing fMRI with optogenetics, and they were showing that when you do optogenetics with the animal in the scanner, you get like a neuronal activity response. But then there was a time where the animal died. It was just anesthetic overdose in the scanner, and then it was during the scan, and then they did the same optogenetic thing, but they were still getting this bold response in a dead brain.
Oh,
And so they started looking into it more, and then a similar thing, like they published the group here published the paper that just the laser shining the light in the brain induces this weird bold response, and all the data that's out there, you think like, how much of that is artifact, how much of it is scary, right?
yeah.
The thing I always say to my students, and to myself, let's be honest, I would say this like a mantra whenever I'm having a panic, which is that it is literally the scientific method to be wrong. Like, you're supposed to throw everything but the kitchen sink at a hypothesis, which means if you're not wrong most of the time, then you're not actually innovating. So so whenever a student comes to me and goes, I did this and it didn't actually work, like obviously there's the technical thing, you go through the list of these at the technical, and then if it's like, what is going on, I just go, well, congratulations. Let's go get a coffee, work out what on earth's going on, but excellent, you've innovated. Like
science right
there. Yeah, because I think we're so used to everything having to work all the time, and especially, the publishing system rewards these amazing success stories, but actually we're supposed to be wrong. That's literally the definition of doing it well.
Yeah, for sure. I think as well, we kind of touched on this at the beginning, but like a bit of culture of being able to say that you're wrong and that something's gone wrong. And we have at the medical school in Dundee, we have these talks called research in progress talks. And I always think the best talks are the ones who genuinely like talk about the research that's happening now and talk about what's working and what hasn't worked. Because sometimes people want to put their best foot forward, and they'll present, stuff that's published actually, because they want to kind of like be like, oh, look, I managed to do this, which is also, you want to have the opportunities to do that as well. But I think these talks in particular are really important to say, actually, I'm having a bit of trouble here, or this isn't working. And when I did my last research in progress talk, I was talking about some of my data. And one of the other researchers from the department, who's not a dementia researcher, he was listening, and he was like, actually, I work in blood, like look at blood pressure, but I do it for something totally different. But I think you should be doing this, and I have this mouse model you could use. We ended up collaborating, and we've had a couple of small grants in because of that. So I think that's a really nice example of when you sort of say about your failures, and someone else comes in and goes, well, actually, or I say failures, let's use failures in inverted commas, failures. Someone comes in and actually there's a door opens down a new avenue of research. So sometimes the best thing you can do is talk about maybe what's not working or what you don't understand in your research the most. So sort of moving back to Ian. So let's talk about the time that an experiment technically worked but answered the wrong question. Have you had a moment where something worked exactly as designed in terms of the method, but you later realize, actually, this is a completely different question that I'm looking at now?
Sort of, yeah. So one of the proteins I work on is called ACPOIN4. It's this water channel in the brain, and the antibodies for it are a bit rubbish because not many people do Westerns for it. Anyway, so I was reading papers, and I figured out that it expresses as these two different isoforms, which you can resolve on a Western blot. And I was like, great, we've got everything set up. Fantastic. You can see where this is going. Western blots are involved. But anyway, so I did this experiment, kind of pulled the proteins apart and could see the two protein bands. But in every single gel I did, there was like this weird hazy band above it. And depending on how the proteins were how the brain tissue was homogenized, it would vary as to how much of this band was, and it made quantifying my protein of interest really difficult. So me and my students spent ages trying to change the way that the proteins were extracted, to change all the things that you change in a Western blot. Literally months trying to get this, because I had this technique in loads of my fellowship like readouts in all of the experiments we were doing. Anyway, so it's during this time, it was kind of the end, and we were like, right, we need to move on because this is, I can't spend too much time on this. And then a paper came out identifying another isoform of this protein. And that's what the band was all along. And I was trying to get rid of it, and everything I tried to get rid of it, I couldn't. But it turns out it's another protein, which I didn't even know about and nobody knew about.
We're laughing in sympathy.
I know,
Right?
I love
I like
it.
that though.
Let's
But-
get rid of
Like, you're doing it
aquaporin
right.
by...
But then it meant that I had all of these, because I tried all the different sample sets I had. I had tried human brains, I tried mouse brains, I tried different animal models. But I had all of this data then. I had all of these Western blot images with this new isoform that hadn't really been described before. So then it turned into, we started looking at expression of this, and then it turns out that protein is quite interesting in Parkinson's, we found out.
Well done.
But it's one of those things where I spent ages trying to optimize something which didn't need to be optimized, and it kind of it was good. We kind of answered a different question with it. We got some data that we didn't expect to get.
Yeah,
So I guess
that's
what tipped you
useful.
off, Ian, was literature, just keeping up with the literature as you were doing your research, which is now a really good example of why you should do that, which is hard because sometimes when you're really in an experiment, you don't have time to keep Googling all the time. But it sounds like you actually found something really cool. So
Yeah, I mean, I'm glad something cool came out of it, having spent 3 months trying to optimize one antibody. But
it makes you wonder about all your little faded grey bands that we see all the time on our Western blots. Like, actually, there might be a whole host of interesting things.
Yeah.
And we just go, oh, background, or oh,
Yeah.
I've done a bad Western.
I don't know when the line would have been, because after that 3 months, I was like, right, enough's enough. And then was Googling around, and then like, oh no, that's why there's another isoform.
Oh no.
Don't know if that happened a year later or something when I would have realised.
Yeah.
Oh no.
Has anyone else had that sort of experience where they've discovered something in their research that they everyone's like,
just like forever.
Did anyone else find a different isoform of a protein? Like,
No,
no,
I haven't found an isoform of a protein, but I have accidentally discovered new chemical reactions by not being able to do maths.
Interesting. Are we talking explosions or
No. I will like to say that I have never blown myself or anyone else up.
Well, that's good. I did nearly get killed in my PhD, not by myself.
Seriously?
Well, maybe that's being a bit dramatic, but my very lovely PhD friend at the time, so we were doing our PhD together, we were in a building that now doesn't exist anymore, not because it got burned down, but because it got knocked down. So don't worry, it didn't go that badly. But we had a sort of L-shaped office research bay. So you'd go in through the lab, and then at the end there was a little office to the right, and all the windows were really high up and really small. And I was working in the office late one night writing, and they were in the lab doing some sterilizing. So they had a Bunsen burner and a nice big 2-liter bottle of ethanol.
People actually use those?
Yeah. They still, they still like old school, like at school.
The classic, yeah.
And then what happened was he kind of knocked the ethanol, and it caught the flame, and then the ethanol on fire went across the bench and then onto the floor. And I just remember hearing him swear, and I popped my head out the office, and there was just like flames on the floor, on the bench. And I was like, I just remember thinking like, this is it. This is how I die. And like, I was so calm. I was like, yeah. And I remember looking up at the windows, and they were so small and so far away. I was like, yeah, no, this is it.
I feel like we do need to check you're okay, but this is also really funny.
And then he was panicking, obviously. Yeah. And then I saw a fire extinguisher, and I was like, oh, get the fire extinguisher. And then he went to get it, and then I was like, I was like, I think the fire flames are getting a bit smaller. And I think we were very lucky in that the ethanol was actually just gradually burning off.
Yeah.
And because it was an old lab, I think it was actually a really well built lab, from like the early 1900s, where they were, genuinely exploding things in the lab on a regular basis. So they made solid labs. And yeah, we really, like, the ethanol just all burned off, and I didn't die. So here's
my lesson to any listeners out there. And I am aware that I'm a bit unusual in that I'm a chemist by trade, do the biology as well, sympathize with the Westerns, but chemistry. Guidance, please, 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 please do not put a flammable liquid next to a naked flame. Hopefully that is now apparent to others. But that is generally not permitted in a general environment of any form. That would not be
part of the fun.
A good idea. I would not back that as an
what?
option.
Sometimes you get a bit complacent, get a bit relaxed in your PhD.
But I mean, there's complacent and there's a death wish.
Perks you up, the way motivates you a bit more in your PhD.
It's only 2 litres of ethanol. It's not much.
Exactly. That's fine.
It was only a wall of fire. Like, it's fine.
I mean, we go through worse every time we send a paper in for review.
Exactly. I'd rather face the wall of fire again than review or two. So well, I'm glad that I'm not at the institute that I did my PhD at anymore, and it wasn't me who started the fire. So, so Kate, this one is for you. Can you share a small but memorable administrative or system-based failure? So, what's gone wrong with something to do with a form, an approval, software, procurement, something that quietly causes chaos that you didn't see coming, and you're like, oh. Admin.
So here's fun, right? So the first thing I thought is when you wrote the briefing was sent through, I spent a lot of time simultaneously laughing and despairing because it was all very clear it couldn't be a big failure, and I was like, small? How do I have any small failures? I'm really not sure. Like, go big or go home is the attitude over here. So I've actually been struggling to find a small failure.
But, go for a big failure then. I want to know about a big failure.
I'm sure we'll get there. So, and then again, that you didn't expect. I tend to expect admin to be difficult. But what I will share is maybe how I spent loads of time trying to preempt a failure, and then the failure still happened. That was what I decided to go for.
Oh,
are you ready?
What happened in that situation?
So,
or is it just...
as I mentioned before we started, that my fellowship ended a couple of weeks ago. So, and I'm sure you guys, have you been to the point at the end of a grant where you actually have to make sure all the money is spent? And I knew that this was coming, and I spent ages, like literally we spent group meetings for about 2 months discussing as a team how we wanted to spend this money. Being like, we need to get this spent, we need to get it organized, we know that it's got to then be confirmed, and then the purchase requisition paid, and that's all got to happen before the account gets shut down, yada yada yada. The amount of, there was even a spreadsheet, and for me that is unprecedented levels of organization. And literally just before we got there, I realized that we are currently, because of the financial restrictions across universities, operating an approval system, which means any expenditure you make over a certain amount needs to go to either faculty or school for approval, and they need certificates, and those have to be uploaded in order to put your purchase order through. So after all of this planning, I then panic going, oh my goodness, we need to get all of these things in. I can't even tell you how long it took to order a fridge. I don't even want to talk about the fridge.
No.
The fridge is too much. But and then I made this big deal to all the teams about having it. And I apologize profusely to my team because I made this big deal about making sure that if you wanted something, get your approval in. But guess who forgot to get her approval in?
Classic.
And then when I did finally get the approval submitted, they went, oh, sorry, no, there's no money left in that account. I was like, how?
I've got a spreadsheet.
The spreadsheet was wrong. Not good. And then I had to scrabble. Yeah, so basically I'd spent so long trying to get everybody else supported, I forgot to do anything for myself. And then it turns out that my spreadsheet was inaccurate because I don't have access to my own grants. You have to request a spreadsheet that comes from a centralized system. So then commitments came on that I wasn't anticipating, that I didn't know about. And then all of a sudden I was like, oh, I can't even have this thing anyway. Okay, what on earth do I do? So it was just a big drama to just spend a couple of $1,000. I'm not going to lie, it was only a couple of thousand.
I think all procurement systems at universities are a bit chaotic. I always remember when I first got my fellowship, they said, they were like, oh, if you want to spend on it, you need to get permission from the grant holder. And I was like, but I am the grant holder. And they were like, yeah, but you need to get permission. I was like, from who? I don't know.
I don't understand. Who was
I
it?
know. It
An
took like a
email
week
to
for them to
yourself?
be like,
I don't know.
Yeah.
Who was it in the
I
end?
was like,
Was it someone like in faculty or something? Or was it actually just
No,
you and they just didn't realise?
no, I don't know what happened, but eventually I was just like, I give myself permission to spend my money. Oh, I actually think admin fields can be the worst sometimes because they feel kind of out of your control. Like, at least if it's in the lab, you're like, oh, I kind of know what I'm doing, or I can get back into the lab or work it out or
It's
try a
just
different
so
approach.
true.
Whereas
It
sometimes,
makes logical sense in the lab most of the
yeah,
time. But with admin, I have neither the skill nor the wherewithal, apparently, nor the fortune to ever be successful in this area. And it's just shown me that normally I'm not organised. And the one time I try to be, I make it worse. So I might
no,
just
I
go
think
back
I
to,
blame the system.
That's a lesson. Yeah.
the
Yeah,
lesson is apparently chaos is the only way I can operate.
I want to pause here and ask all of you. So, why do you think we are so bad at talking about these moments in research? You know, is it, I think it just comes back down to sort of culture again, where people are just scared to admit failure in case they get in trouble. But actually, I think some of the best, like certainly in research and moving research forward, I do think the best thing to do is to talk about failure, because a lot of time there might be someone in the crowd who goes, actually, I know why that's failed and it's really interesting and you should look at X, Y, and Z instead. Obviously, we're not talking about like, oh, I dropped my ELISA before I read it. You know,
Oh,
something
split my
like that.
gel.
Classic.
Split my
split the
gel.
Gel's
Splitting
gone.
gel's fine. You just kind of like smush it and then hope it's still
Squad it back together.
exactly. Just
Glue
keep
it back together.
Isn't that how you
It's fine.
rebake a cake? Just chuck
And
some
gels,
sprinkles
it's all
on
the
it and
same.
cover the cracks.
It's all the same.
Yeah, I mean, I think everyone else can sort of,
Mycoplasma. Sorry.
everyone else can
fear
sort of
into the
say,
heart of everyone on the podcast.
it's fine, we'll edit that out as well. It's Christmas, not Halloween cake.
Ew.
So
Actually, I was going
to
to say on that note though, with them kind of being quiet about stuff, one of the things that I think really annoyed my master's student this year was that I was always like, I don't really care what's happened, I just need you to tell me, like, because otherwise I'll come into the lab like 3 weeks later and something will be broken, and I like, that's more the thing that annoys me. So I really don't care if anyone breaks anything in the lab, or like, I don't know, chuck something away. Like, I've had students, like you said with the ELISA, just tip it out at the end and I'm like, no, that was but anyway, it's fine. We take a breath, we start again. But I actually get really annoyed when someone's like broken an objective or something. You know, a month later you go to use the microscope and it's broken and you're like, okay, so now I have to figure out what's going on. I have to start the repair process again. None of it's a big deal. It's like a month later and you're like,
and if you know,
Absolutely.
like, our jobs are to fix problems, right?
Yeah.
And it's to try and fix scientific problems at the end of the day, right? So if somebody comes to you and say like, I've cracked this piece of equipment, you can be like, that's fine, and I can fix that. I can buy another one. But then going into the lab next month and being like, oh God, okay, well now I can't do my experiment because, yeah.
100%. Yeah, absolutely. I think, like, I mean, I've broken stuff, and if it's not mine, what I usually do is order it straight away, go to the person and say, listen, I've broken this like thing, and
That's very
I've ordered it.
conscientious of you.
You've got to do that though. I mean, that's,
I mean, yes,
you know,
but it's nice.
yeah, I mean, not, I guess, like, I think sometimes people are scared to say if they've broken something and they just try and hide it, especially, you know, if they're not permanently in that lab and they're like, well, I'm out of here soon, so we'll just cover it up. But yeah, I always say to my students, just, you know, it's never as bad as you think it's going to be. You know, it's never that big a deal. It might seem it at the time, but nothing's that big. Nothing's that big a problem that it can't be solved. There was another situation during my PhD that I thought was quite funny. Was we moved buildings. So as I mentioned, there's an old building that's not there anymore. We moved to a new building, and it was getting opened by Camilla, the now Queen, and she was going to do a tour of the labs, but we were already in them. We'd only been in them a couple of weeks or something. My good friend, who might have been the same person that started the fire, they were filtering a thing called oil red O. It's a lipid stain, and Ian's nodding, he knows what it is. So, I mean, it does what it says on the tin. It's very red and it's a stain. And he was a bit impatient when he was filtering it, and he had it in a big syringe. And he just was like, "Oh, I need to get this filtered faster." And whacham, hit the end of the syringe, and the whole thing exploded. And so this oil red O, it went everywhere. It went all over the shelves. It went over the top of the new, like brand new lab benches, and went all over the floor. And of course he and himself actually, like, he came out of the lab and it looked like, I thought he'd hurt himself because he was covered in, like, his face was covered and spattered in red. And I was like, and his lab coat was covered in red. I was like, "Oh my goodness, are you okay?" Like, I thought he'd like cut himself or something. He was like, "No." So we saw the lab, and like, it looked like a murder scene. There was just like this huge pool of red on the ground. And so we tried to clean it up. But I don't know, like I say, it's a very new lab. I don't know if they properly coated the floor yet or something, but it just literally rubbed into the ground. And so Queen Camilla was visiting like the next week or something, so they tried to get professional cleaners in, and like literally nothing could get this stain out. So eventually they just had to like just, when they were giving the tour, they're like, "Oh, don't go in that lab. We don't need to see that lab."
No, this way.
Which I feel that if she then had seen the state, like all this red, she would have been like, "What happened?"
I don't know. She probably found it really funny.
It was, honestly, it was so funny. Yeah.
Can I ask a question, Fiona, please? Is this friend that you speak of, are they still in research?
Yes, and they're a very good researcher.
Could we please bring them onto the podcast? I want to meet this legend amongst men.
They're still, I think, they're still a researcher. They're very good scientists.
Oh, I'm sure. I have no doubt.
Just a little bit of, like, that's what I say, you learn by doing. And he never rushed filtering, in particular oil red O, ever again. And I never rushed any filtering ever, because I've seen it. And like I say, as long as you learn something, and nobody actually dies, then it's fine. But anyway, moving on from that story. So I guess we were kind of talking about, why we should talk about these moments. And do you think it often links to imposter feelings? People don't want to admit stuff because they want to, especially ECRs, early career researchers, they want to prove themselves and be like, I can do research. Sometimes some of them are trying to prove themselves to a lab to stay on as a PhD student, or they're about to finish their PhD and they're looking for a postdoc position and they want to impress people who can potentially hire them. Ian, what do you think about this?
Yeah, it's a good point, because I think a lot of academia is engineered such that it's all about kind of putting your best foot forward and like selling the papers that you got. It's all about putting these personal statements together when you write fellowship applications, for example, saying how amazing you are and what amazing research that you're doing. And so we're so trained and it's so ingrained in us to point to all the amazing things about ourselves, rather than those, and how many of those Western blots go into the paper, all of the other ones that you did and all of the other data points that didn't quite make it, or you were optimizing. But that's not the thing that's seen, is it? And it's in every experiment you do, there is always failure to get to the one where it works. But I guess I think it's just a failing again of academia, isn't it? It's just, we're trained this way, and we should be more, podcasts like this when we're actually talking about it. And Fiona, what you said earlier about having those kind of work in progress sessions, like, they're so good, because you put up your actual data that you got that week, warts and all, and say this is a rubbish immunohistochemistry stain I did, look, and I'm trying to do this and I'm trying to do this to talk about how you're trying to fix a problem rather than just, showing the polished version at the end.
Yes. And that's where it can save you both time and money, because if you're constantly showing what you're doing and being honest about what's not working, rather than just maybe showing the bit of the experiment that did work, that's where someone can step in and say, oh, I know why that's not working. Or actually, you're maybe not interpreting this quite right. Here's how you should interpret it. And you can move forward doing this instead. I do think in academia, like any talk I've given, I mean, very rarely does anyone, get to the question section and say, everyone in the audience is like, brilliant talk, no questions. Like, I mean, it's always people always, people always say, actually, have you thought about this? Have you done this? And it can be, as you say, a very critical environment, academia. And it's kind of about, I think sometimes just embracing it. You can be critical in a nice way, though. I definitely think
100%. Yeah.
Yeah, it's to do with tone and how you ask questions a lot of the time. That's why my PhD supervisor always taught me to sort of, if you ever get quite an aggressive question in a talk, you always say, oh, thank you for your question, or that's a really interesting point you've raised. And it kind of like, it gives you a bit of time to think, but also just maybe chills out the tone a little bit if someone's sort of saying, actually, I don't like your research, it's got this wrong with it. So, well, I hear your point.
Sorry you feel that way.
I'm so sorry you feel like that. You are wrong. Soraya, you're laughing. You can maybe.
I am. Again, when I was reading through this, I was like, I don't make mistakes. We're all good. And then I've done just because, like I said, I don't see them as mistakes. Well, I have mistakes as well, but they're all learning lessons. But I never try to schedule any experimental stuff in like the week before I go away. And I've done 2 this week because, of course, why not? And both times I've done something wrong in them. And I was like, this is why I don't do stuff this week.
So do you think it's the pressure, or do you think it's just bad luck?
I think my brain's a bit switched. I don't know. So yesterday I was showing someone how to do an ELISA, and it was kind of the only time that we had like over this month, because I had a conference the week before and stuff like that. I was like, we'll just do it nice and chill, no rush. We'll just read the protocol. We'll go through, blah, blah, blah. But I'd actually drawn my little well plate template wrong. So I'd only given myself 8 columns rather than 12 columns. So then we'd processed all the samples, we were loading up the plate, and I was like, oh, we have another 4 rows on this ELISA, and that's money that's going to go down the drain if we don't use it. So then I was like, right, you keep loading. I'm just going to start doing dilutions off the back of my glove to try and like rescue this. The dilutions didn't work. So, I mean, I should have just left it as is, but I tried. The rest of it worked. The 8 worked. It's just the other 4, just money in the bin.
the classic back of the glove, like, oh, let me get my pen, and or the other one is the paper towel.
Yeah.
The paper towel roll.
Yeah.
Blue roll. This will be fine. Like,
do you guys
wonder how many Nobel Prizes have been written on the back of some blue roll or something?
Memo, post-it note or something.
Post-it note. Oh yeah, absolutely.
Do you guys ever find out? So when you're like, you've planned an experiment. I have this thing, and I wonder if anyone else identifies with this. Get excited because I've got 1 million cells, and I go into it going, I'm only going to do these experiments. I say 1 million, I'm talking more like 24 to 30 million, but loads and loads of cells. And I've gone, I'm only going to go in doing these experiments. I'm going to be sensible. I'm not going to create chaos. There's no need to push the envelope. And then you suddenly see that you've got 99% viability and you've got a really good number of cells. And then out of nowhere, you go, I'm just going to do another 10 plates. I'm just going to invent the experiments on the fly. Let's see what I can do. And then you end up massively regressing it.
Yeah.
And it never goes as you think it's going to go. Never goes as planned.
Does anybody else have this? I cannot control myself.
Yes.
Always. Just add another antibody on. We'll just look at something else as well.
You know what?
I
wanted to try this random experiment that I've never tried before. I'm just going to see if I can do this with a completely different cell line. Why not? Even though I've got loads of these cells.
Yep, absolutely. Yeah. I think, Soraya, what you said about the antibody, if you're adding 1 antibody in, why not add another 20?
One? There's enough space on that channel. Let's forget about the fact that they bleed into each other, and after a while they cancel
out. That's always that's it.
It's the red and the green.
Yeah.
Why do we do this?
I had a similar thing with an ELISA, which happened,
Classic.
and it almost repeated itself literally last week. So the week before, I think it was before my fellowship application was due in, and the last bit of experiment, and I just needed to show that I could detect alpha-synuclein in CSF, and I had the samples. I ordered the plate, and I was waiting for it to arrive, and then it arrived on, I think it was the Tuesday or Wednesday before Easter, and I was like, I just need to do this ELISA. It literally got it out the box, it got delivered, and I went straight into the lab to do it. Obviously, I was rushing it. I didn't read the instructions properly. I put the wrong concentration of the antibody in. It didn't work. And then so my fellowship application was, I had a little gap where I was going to put in the graph. And so the Easter was spent rewriting it and getting rid of that figure and stuff.
I mean, you still
but
got it, right?
yeah, it's true. But literally this week, the same thing happened. I needed to do these surgeries, went to the freezer, figured out I didn't have enough of the tau brain homogenate to do it. And I was like, right, I need to order an ELISA and I need it now. And then I need to quantify it so I can do the surgeries next week. Ordered it. Inevitably, it got stuck in customs in Germany. And then it eventually arrived. And then I literally got it out of the box, came straight into the lab. And I was like, right, Ian, take your time. read the instructions. Take your time. This cannot fail.
He's making a list.
But it works and it's fine. So I've learnt my lesson.
Yeah, I actually think like 96 well ELISAs aren't 96 well ELISAs. You have to give over. I probably don't do it as well, but yeah, you have to optimize what concentration, like of dilution of your sampling you need to use. I always try and do it in 2 strips, so like 1 strip for the
doesn't go
the
as far as
and
you
1
want.
strip, and 1 strip for the different dilutions of like a couple of samples. And I've been lucky so far in that's worked, but
it's a gamble.
I mean, I had recently. I wanted to add more. I had that recently. I wanted to add more samples to. I just wanted to look at more samples. I looked at using an ELISA, and I ordered the same ELISA. And what I learned is that all ELISAs are different, even if it's the same ELISA. If you want to compare samples, you have to do it on the same plate with the same standard. And what I really worked out is actually the standard was different. It wasn't my samples, because I was like, why is this skewed? I did one of the same samples from the last plate on the new plate just to double check consistency.
Very nice.
And it was the standard was off. And luckily I actually had one like some old standard left over. I put that back on the plate and ran, and it was then like the old standard. And oh my goodness, sorry, I'm saying the word standard a lot. Basically, the word standard doesn't means anything but standard, because they were all different. I feel your ELISA pain.
Whilst we're on the subject of absurd assay-based situations, how would you feel, Fiona, about me sharing a pretty gargantuan fail related to an ELISA, so that our audience hopefully won't lose faith in any of my brain cells, but potentially, but also will go, oh my goodness, this person has managed to get a senior lectureship and they have dropped the ball this severely.
Go for it.
Would you like
ready.
that?
Yeah.
I please reassure you, listeners, that it's not going to seem it over the next couple of minutes, but I am competent. I'm going to caveat this with, it came over a time where I was first heavily pregnant, then postpartum, then heavily pregnant again. So it was being raised to me at a time when I had 0 brain capacity or Braxton Hicks. That is going to be my defense. But
I feel that's a fair defense.
So basically, we're on a project where we're using phenotypic screening to find new anti-neuroinflammatory drugs. And we have our beautiful report line that works really nicely. We've spent 3 years setting up all the orthogonal assays, so we know exactly what it's telling us. We're not doing this half-baked. And we have set it up with our Z prime, our signal to noise. It's the kind of work that an interdisciplinary scientist would be proud of, and things were going really, really well. We'd even found new compounds with a new mode of action and all this kind of stuff. And then I remember about 2 years in, my students saying to me, Kate, is there a problem? Because I've noticed that when I've been taught this assay, the guy in the HTS facility is putting the compounds on for 2 hours and then taking them back off before running the assay. And I don't know whether I didn't really understand what was said or whether I just didn't have the capacity to process what was said. But I went, just make sure it's consistent with what you're doing. Right. didn't think much more about it. There was a little bit of, oh, how would we interpret this? As a chemist, you'd leave those on, but maybe as a biologist, you'd take them off. And I do a lot of thinking it's because different disciplines or whatever.
Depends how they
Fast forward a couple of years, where my next student is working on it and learning to run the assay, who goes, Kate, and this was when I'd finally stopped feeding, so this was only a few months ago, and was no longer pregnant and had some sleep because my daughter's now 18 months. And he went, Kate, you know that the protocol as we currently do it has us putting the compounds on that we're dosing for 2 hours, then taking them off and washing them off, and then running the assay to see if the compounds have any effect. And I was like, care? And it was like the first time I'd heard it, and I was like, this is not the first time I've heard this.
No.
And it was like ringing in my ears.
Like your brain needed it twice.
And I was like, what? So there's no compound on the cells when we're running the assay. But we've run several hundred analogues through this at this point. And then we were like, no wonder we're not getting any structure-activity relationships.
Oh, no.
I told you, this is idiotic of me. And I was like, what? And I think it was, I couldn't possibly believe that would have been, but because I hadn't run the protocol myself because I was off on leave, I just was like, obviously you'd leave them on. And I don't know whether I just didn't process it or whatever it was, but I was like, no. And then I remember.
To be fair, like sometimes in assays, like you do treat them with something, remove it, and then put the drugs on, or vice versa. Like it does happen sometimes.
But it's we were stimulating after putting the drugs on, leaving them for 2 hours and taking them off.
Oh dear.
God, yeah.
So to be fair, the fact that we got any active compounds out of this assay at all, I was like, oh God, those are going to be good, aren't they?
They must be really good. I was going to say. Maybe that's the way forward if you want to find something that really hangs around.
I mean, it must have been some good binders. I'm not going to lie. We were laughing our heads off. Now I can laugh about it. But at the time, I was like, I should quit my job. Like,
no.
What? It's so embarrassing. And then it just never computed. And then we had to go to our collaborators and explain that this had happened.
Yeah.
And they were like, how did this happen? And I had to just be like, I can't style this out. It rests with me, but my brain was elsewhere.
So I guess that maybe leads me on to my last point is there anything in a sort of situation where you sort of see it as a failure or a fail moment that you'd maybe now do in a completely different order? So I guess, Kate, you'd maybe run that protocol in a mini way in the lab before scaling up.
What I do in a completely different order is wait until my brain is functional before adopting a new protocol in the lab.
That's a good point. Yeah, I get it. And you do have to pick when you try something new in the lab, because you have to be in the right mental state and I wasn't. Have the energy for it and the time. As Ian said, he rushed his ELISA. But yeah, is there anything that you've sort of learned? I do something in a completely different order now.
No, I think just like you say, just giving yourself the time, giving yourself the peace if you're going to try something new. And just, making, I don't know, as we've been going through, I'm just like, there's so many fails. I'm not just like so many mistakes.
Learning experiences.
Learning
experiences.
Sorry,
Don't
learning
lose your
experiences.
Sparkle.
So I've got two quick anecdotes very quickly. Is that one, both colleagues of mine, one of them was like trying to show that tau spread in flies and thought didn't have his secondary controls right. So then there was no tau spread in flies. So then he was like, oh, boo. Then he tried for like months to get tau to spread in flies, didn't get it, but then published it. So that's a learning lesson I wanted to add here, is that sometimes if you're like, it's really not working, maybe it's really not working, so then you can publish it. So that was a good little outcome.
That's often said, is people need to be able to publish their
brain communications.
Nice.
Very supportive of negative data.
I'm just going to write that down.
For all of you can't send everything that you failed at. It has to be like
Can I not? I just wanted to send them my diary.
But it's such a good point. Like, people don't typically publish their failures, but it would save so many people so much time.
And actually, he got so many like emails afterwards to be like, we've been trying this for months and like it hasn't worked. And
and you always know the best papers always reply. this was something that worked, but actually when I worked on a Huntington's project, it was quite difficult to genotype the mice. But there was a really nice, oh no, I'm absolutely lying. It was my DB line. It was a diabetic line. Told a lie. It was difficult to genotype them. And this group had published a paper on a sort of a way to do it. And it was really interesting because you had to use like a really cheap version of like the chemical that cuts all the bands up. It couldn't be too good one. It had to be a rubbish one. And I loved that. It had to be like a messy one. But anyway, the point was is that I emailed them because I needed some help. And they emailed back immediately. And I was like, you know the pain. You know what we go through that same pain. And I love that group. They saved me so much time. But anyway, sorry, you were saying, so you had a second and
I to be fair, the second one is also just an absolute pain for my other colleague, which was he's been hired to like look at this protein, has tried many antibodies. So when you said one antibody, I was like, oh, at least it's one. This is like thousands of pounds of antibodies. He's gone through the ELISAs. He's now on RNA scope, and that's roughly working. But we went to the Scottish Neuroscience Day actually, and one of the people stood up and gave a talk and was like, this protein group is notoriously difficult to detect. And his face just went, just like completely dropped. And he was like, yeah, tell me about it was like, it's not really a mistake. Like you have to be confident that you're doing things right. But science is so full of like mysterious turns. And
if science was easy, everything would be cured and everyone would do it.
That's true.
That's why I always say you have to be slightly obsessed with whatever your topic of research is to want to do it, because it's so tough. But you get those highs. I imagine
it's like a drug addiction.
Work, you get this high. You're like, oh my goodness. Or if you find something, it's like, oh, it's addictive, that feeling. I think that's, and those are the people who stay in research, the ones who sort of keep.
If the highs outweigh the lows, it is an emotional roller coaster.
Yeah. But you do get more lows and highs. So
I did text. Literally, I texted a researcher friend yesterday because we're in the same institution, but like I think she was teaching and I was running around doing something, and I just literally went, in the space of 24 hours, I've gone from profound despair to total elation. What is this job? It was literally just based on, I think the day before I felt like I was never going to manage as a researcher and I should quit, and then it literally had gone, just because someone I thought was very important liked one of my ideas, to like, I'm on top of the world. I was like, this is exhausting. I think we need a Christmas break now.
I think that's where like compartmentalizing things is really useful. And I guess that sort of brings me on to my last point is there a research fail that you can now laugh about? So Ian, have you got something that you think back now at the time was really bad, but now you find it quite funny?
I think I'm just about find it funny now. This example so there was we were doing these crazy experiments where we were injecting a contrast agent in right at the back of the brain. So it's like this horrifically complicated surgical setup, and we had to do it in the scanner bed, get everything set up, and then move the scanner bed into the MRI room, into the MRI scanner, with all the cannulas and all the wires and everything coming out. So it took us forever to get it running. And then, so you do a baseline scan, then you start your infusion. And then I used to check the first few scans to make sure that I could see contrast. And then you can kind of be like, right, okay, fine. Now we're imaging for 3 hours. It's fine. So the first few, once we, Soraya's laughing at me. Once we got this running, I was like, right, now I've got everything optimized. So I just stopped checking the first few scans. And then it turns out that the memory on the MRI scan computer was ridiculously low. So it was a couple of days in. And then I was like, oh, maybe I'll transfer some of the data over to my laptop and then I can start doing some analysis. And so there was like a good 2 or 3 days of scanning that the data just hadn't saved. And I was like, well, that's time I'm never going to get back, isn't it? And then I tried to, but somebody was booked on the scanner the next week, and I was like, oh, okay. Well, lesson learned.
Oh,
now every time I scan, Like I take the first scan off, like reconstruct the data. I'm like, right, it's there. This is how much data I've got left. This is what the quota is in the system. I was like, okay, I'm good to
my
go.
goodness.
Every
Poor
single time
thing. I'm
without
just
fail.
imagining you
But
as some kind of like little data gnome, just like, I must have my
you're like,
data. Don't take it from me.
Oh,
I'm so sorry.
bad times.
That is painful. That is very, very painful.
Oh.
But you said you can almost laugh about it now.
Yeah, I mean, you know.
But again, you learned something from it.
Learning lessons.
Exactly.
Learning lessons.
Not fails.
And Soraya, is there something that you can now laugh about, something that happened to you?
Yeah, I'm going to keep this narrowed to this year. Like a lot of these are basically this year as well, to be fair. We have a lot of fails. So going back to Millie again with her master's project, she was doing this ELISA, because ELISAs are the devil now that we've started talking about them.
Yes, agreed.
And we'd got a new batch, and the protocol was very similar to the old batch. And bless, like Millie was like, oh, you know, it says here that we need to shake it for an hour. And I was like, we've never shaken it before. That doesn't matter. Like being the, you know, the experienced ELISA runner that I am. And then it just didn't work. And I was like, what the hell's going on? We thought there might have been problems with the water. So we were like getting in sterile water from different places. I'd emailed the company to be like, guys, this isn't working. Like, what the hell's going on? You know, can you send us a new kit? Because these are expensive. And then like, he was like, and actually, I think they must get this a lot, because he sent a very detailed thing back to me, like, have you done this? Have you done this? Have you done this? Check this, check this, check this. Because we got no signal whatsoever. And I was like, what's going on? And then, I was like, this works. Right, I'm just going to follow everything exactly as it's... Oh, it's worked. Okay.
Oh, no.
The shaking doesn't work.
You have to send that emaal back saying, hi.
I didn't email them back. I just kind of ignored it.
Power play.
Ignore that.
They already know. They know that I've been an idiot. Like, I haven't followed it up. I don't
They'll
need
see another
to.
order from you for the same plate.
Exactly.
And then, she's got it working.
Do you think they might just give you one extra free?
Well, I was kind of just like, oh, this kit's obviously not working. They've done something weird. But no, it was me. I was the weird.
No. And Kate, is there something that you can now laugh about that happened to you? Can you laugh about your big drug screening assay?
Probably not.
It's still a bit raw, I'm not going to lie.
Raw.
But what I will say is it's along similar lines of what Soraya is saying. So this isn't a biology one, this is a chemistry one, but it's along the same lines, which is you end up sometimes when you do a chemistry reaction, so if you're making analogues, something doesn't work, which is quite common. First time you do a chemistry reaction, it often doesn't. Optimizing every single tiny aspect of the reaction, being like, okay, I need to try different catalysts, and you can literally spend months re-optimizing a reaction. And then it's one of the things that theoretically you should always do, and you know that there are similar practices with assay development. You know, you should always check your housekeeping genes, etc., etc. We don't, and I know that we should, and it comes back to bite you every once in a while, and then you remember. And one of the things in a chemistry, I don't know if you guys have come across this, is it's always a good idea when you're ordering a new chemical to check it's actually what's in the bottle.
Classic.
And normally you just trust it. And then you go back and then you NMR it and you're like, oh, that's actually nothing. That's actually just, I mean, there's not even a chemical in there. It's just mush.
Oh, no. See, in biology, we just trust that what gets sent.
Yeah, I've not even thought about. Yeah.
No, and this is probably why I'm quite annoying to purist biologists, is that I tend to get very critical of kits. I don't trust anything.
That's fair. Yeah, I mean, I wanted to look at iron in tissue, and I ordered a kit, and I wanted to look at it in brain, and it said that it could do tissue homogenous. So tried that, and it just didn't work. It couldn't detect anything, and I think they'd only tried it on liver and gone, liver's tissue.
That'll do,
That'll do for everything. But what was quite nice about that is it actually led to a really cool collaboration with this group of people called the UK Measurements Laboratory, who they measure stuff. They're amazing. If anyone ever wants to measure something, I know that sounds really broad, but they measure stuff. Then they're great to work with. But to find them, I ended up like cold emailing like the most random people in the UK being like, can you measure iron? Like just literally to like physicists and random academics.
Help
and stuff.
me.
But lovely physicists, lovely community, because they were like, these are the people you need to work with eventually. And I was like, Mary on a donkey looking for Bethlehem, but instead of Bethlehem, I found the UK Measurements Laboratory. I'm trying to bring in the Christmas theme.
I wondered what was going on here a little bit.
Pulled it back. I see. Yeah, it's good.
Pulled it back.
I was like, okay.
So I think on that,
great job.
Terrible.
That was amazing.
Anecdote.
Never change.
Thank you so much to Soraya, Ian, and Kate for being so generous and brave enough to share your stories. And if you are listening and you have had a messy research moment this year, you're in very good company. And we would love to hear about your own research fails of Christmas. So do share them with us in the comments or in the community app. And I think on that note, I wrote something down that I quite like that Soraya said earlier. And she said, give yourself peace to do new things. So I thought that was my other Christmas thing, peace on earth, or in this case, peace in the lab. And so I'd like to say thank you for listening, and thank you for being part of Dementia Researcher community this year. Bye!
Bye!
Everybody say bye.
Bye!
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk